Today, I will discuss evolution and development from the point of view of how we acquire human knowledge through the visible and the invisible. I will divide my talk in three parts. First, creation and evolution. Second, the discovery of development controlling genes. And finally, how natural selection created these beautiful forms of animal diversity using these conserved genes. The problem of creation has interested theologians for all religions for the longest time. And Pope Ratzinger, Benedict, wrote a little book called In the Beginning. And in it, he discussed science and religion. And the end of the book says like this: only if the Redeemer is also creator can he really be the Redeemer. Now, the best explanation I know doesn't come from a theologian, but rather from our previous president, Werner Arber, who wrote, the chapter of the Genesis in the Old Testament is for me a testimony of an early um, scientific view. Sorry. Of an early scientific worldwide view already existing several thousand years ago. This chapter also reflects a wide consistency between religious faith and available scientific knowledge. It proposes a logical sequence of events in which the creation of our planet Earth may have been followed by the establishment of the conditions of life. Plants were then introduced and subsequently provided food for animals before human beings were finally introduced. This sequence of the earth, the oceans, plants, animals, and humans is what science believes, except that instead of 6 days, there were millions of years involved. An important event in this world of the visible and reason came in 1830 in the famous uh, uh, debate between Geoffroy Cuvier and Geoffroy in the French Academy of Sciences. Geoffroy, shown at the top, dissected a lobster and said, what was my admiration that if I turn around this lobster, then the nervous system will be on top, the heart on the bottom, and the gut in the middle. So he lost the debate, but this idea of a unity of plan struck a chord with philosophers and writers, because science and philosophy were not separated in 1830. For example, Balzac wrote, there is only one animal, the creator has used only one pattern to create all animals. And it, then, this was 30 years before Darwin. And Darwin realized the potency of natural selection. And he said, essentially, the theory is that natural selection of spontaneous variations drives the differential reproduction of individuals. Reproduction is copious, as proposed before by Malthus, and the struggle between biotic competition serves as a creative, according to him, mechanism throughout the periods of geological time. And so this is more or less completely also what we think, but except that there are catastrophic events that they are more important. But this is from the world of the visible and human reason, but then comes from the invisible, 100 years ago, from genetics and from embryology. 100 years ago, Thomas Morgan found a fly that had 4 wings instead of the normal 4 and called it it's a homeotic transformation, that is the transformation of one part of the body into the likeness of another one. And This mutation was studied for decades by Edward Lewis, and he found that it was a deletion of many genes, shown at the top of the slide. And then when and proposed they would be duplicated, and when Walter Gehring in Basel cloned the first homeotic genes, he found that they had a very conserved region looking for the duplication. And it's very conserved as a segment of only 60 amino acids called the homeobone, that it's so conserved because it fits perfectly into the helix of the DNA. And I was at the time in Basel, and we cloned the first vertebrate gene using the 3 Drosophila probes. So there are those black bands there. And we said at the time, a conserved gene was cloned on the basis of cross-homology to a region conserved between several Drosophila fruit flies homeotic genes. These genes could perhaps represent the first development controlling gene isolated in vertebrates, and it was. For when we obtained the antibodies to see this gene in the mouse, we could see that it's expressed in 8 segments of the thorax, something that you would have never predicted. And through the work of many, many labs, certainly not ours, it was found that these genes, homeotic genes, or HOX, are present in the mouse or the human in the same organization as they are in the fruit fly. For example, the red gene is the most anterior, and the green gene is the most posterior. Now, if we look at the fly embryo, we know if you look at the left-hand column and you look down, there is a blue gene, then a green gene, magenta gene, turquoise, orange, and then red. And they are like stripes, that it's like a salami, the embryo. And if amazingly, if you look at the similar genes in the fish, you also see the same. For example, if you look down the first column, you see a blue, a magenta, and a green that are increasingly more posterior. This led evolutionists to say this was one of the finest fishing expeditions in human history, and it took 40 years ago. The important second event came from embryology. 100 years ago again, Spemann and Mangold did this experiment, here repeated by me, in which they took a small region of the frog embryo, which they called the organizer, that you can explant it and transplant it into a host embryo, as we're going to do now. And we make a little hole on the embryo, and we do it all this just freehand, and we put it in the freehand, and we put it on the little bit of the embryo, and give it a little push, and this heals perfectly. And 2 days later, you get a Siamese twin, in which the transplant just gave rise to the notochord, but the segments of the muscles and the whole brain and the eyes are induced in the host embryo. And so therefore, when cloning became practical, we and others took that little fragment of tissue and cloned many genes, of which the most informative one was one called cordon. And cordon is an inhibitor of another growth factor called bone morphogenetic protein, and it's expressed on the dorsal side, meaning the back side, and it diffuses all the way the protein to the ventral side. And this generates a gradient of BMP signaling. What you see there is an embryo of 10,000 cells. Each dot represents a nucleus with varying amounts of BMP signal. And we know that to get this, you need a whole set of genes, of which one colloid is a protease that digests cordon, making maximal BMP signal on the ventral belly side of the embryo. So this is very important because it determines the tissue types, such as notochord, somites, kidney, and so on. But it also determines body shape. And as most of you that were children would have seen these, double-twinned goldfish, which are change the body shape very much, and they are uh, uh, a mutation in cordon. And so this system of dorsoventral is shown in A for the frog and in B for the fruit fly has been completely inverted, giving strong support for Geoffroy's theory of the inversion of the lobster. And this system has been kept from the frog and humans all the way through the flies, the spiders, and many, many animals. So we can say that the cordon, toloid, BMP pathway is a molecular machine used to establish a diffusion gradient spanning the entire diversity of animal embryos. And this then brings us to the important observation that both the invertebrates at the top or the vertebrates at the bottom come from a common ancestor that lived in the bottom of the sea called Urbilateria, and that a little animal had 6 OX genes in a row, and it had the complete BMP cordon toloid system in place. But it's even more than that, because both in the protostomes, which means mouth first, and in the deuterostomes, which are mouth second, there are primary larvae that live in the plankton of the sea, and they feed and they disperse the species. So this animal likely had a primary larva in the plankton, then the adult form settled at the bottom of the sea. And so the evolution of animals of Earth has followed a very thin chain of most unlikely events. Many would even say miraculous. So we think that an ancestral network of genes was used to build the beautiful variety of animal forms under the strictures of natural selection of the fittest. The obligatory of use of ancient developmental gene networks channel the almost miraculous evolutionary outcomes. And what is important is not all animal forms were possible. They had to be compatible with the genes of embryonic development and with the laws of physics and of chemistry that were set up at creation. Next year, we will hold a evolution and development workshop here, and I'm happy to say that ours is the only religion with an Academy of Sciences. So thank you very much.